Well, good evening, and welcome to Chapter 26, the urinary system. This is Bio 212 from Norwalk Community College, and what we're going to do here is we're going to break Chapter 26 up into two or possibly three different pieces, and we're going to look at first the structural regulation or structural requirements, and then structural requirements for purification of the plasma of your blood and interstitial fluids, so that we can do as I have right here on the sheet for you. You can see regulation of blood ionic composition, regulation of pH, regulation of blood volume, regulation of blood pressure, maintenance of blood osmolarity. We're going to use the kidneys as sites of synthesis for calcitriol and erythropoietin, as well as regulation of blood glucose levels. And even though we don't get into it in terms of foreign substances such as drugs and toxins, excretion of wastes. You remember, if we are making carbon dioxide as a waste product, that means we're going to have a lot of hydrogen protons inside of our body. And really, at the end of the day, you got to figure out a way to keep that pH balance in range as a result of the waste from metabolic reactions. So, the histology, we saw this inside lab, but let's just make sure you can draw a quick example of one here, right? So let's just say this is the kidney, and you'll notice it has a specific shape to it, right? And there is your ileum here. The renal ileum is where the ureter emerges, okay, along with blood vessels, lymphatic vessels, and nerves. So we just change colors here. I'm just going to put sort of orange yellow. Oh, it's going to be yellow. Look at that. And what's going to happen is we're going to collect materials. You can't see that. It's useless color. Useless color. Let's see this one here. Let's see if this one works better. There we go. Materials are going to be filtered and collected and sent out of the kidney. Notice what I'm doing. I'm demonstrating to you that essentially the kidney is shaped, okay, so that you actually have regions where purification or filtration are going to occur. You'll see how that happens. But before we get to that, there's a capsule on the outside which maintains the shape of the kidney itself. Let's go back to our normal color. So that you can actually have our sort of dynamic space. You need to increase pressure inside this. You've got a capsule that holds it in place. You need to decrease pressure inside of this. You have something that can withstand those changes. Okay? You also have an adipose tissue layer, which is a mass of fatty tissue that's going to protect. Put some sort of dotted lines for you here. That's the adipose tissue. This is going to protect from trauma. Pretty much, okay? And then the renal fascia, okay? It's thin connective tissue that anchors the kidney to the surrounding structures in the abdominal wall. So literally, something is going to hold these things in place. Remember, this is retroperitoneal. This is behind everything inside your abdomen. What do they look like inside your body? Let's get out of this first. Let's go here. And inside your textbook, you can see this figure here where they actually have all of that for you. Here are those arteries, veins. Here's the kidney itself. Here's the suprarenal gland, and you know that is the adrenal gland. Okay, and you're going to see here where the ureter actually comes out, both right and left. Most people are born with both. Some people are born with one. One's malformed, sort of, it was like compressed on one side. Where it is, as I told you a moment ago, it's retroperitoneal, and you can begin to see those shapes I was telling you about, where we're going to figure out a mechanism that's going to be required to actually filter the materials that are passing through your blood. Okay, so how do we actually do that? That outer cortex, so let's draw one again. Let's go back to this. Let's go here. Let's go here. I'm going to make this one slightly larger. Okay, and you're going to see that we have a cortical region. Gee, this sounds familiar, right? We seem to have a cortical region in everything we're studying this semester, right? And then you have a medullary region, okay? So the outer cortical zone and an inner juxtamedullary zone. This inner line here will be the juxtamedullary. So this whole thing here is in white. The whole thing is white, right? That's all cortex. And what we're going to do is we're going to take areas and we're going to put them through And we're going to create these things that are called columns. Okay, and inside, in between each of these columns, we're going to have these things that are called pyramids. Okay, whoops, not what I want to do with that. Let's go like this. There goes my cartoon. Oh no, my renal pyramids are gone. Renal pyramid. Control, underline, bold. Right? And each pyramid, okay, has the ability to actually do part of the work we're going to take a look at in a minute. So how does this actually happen? The functional portion of the kidney is called the parenchyma. Okay, it has within it a series of nephrons. I'm going to put that there. An individual filtration unit is a nephron. We have a few of them. We have more than a few. And they are the functional units of the kidney. And what happens is as urine is formed, it's going to drain into the ducts, which will then be received by the minor to major calyces, which is then received at the urinary pelvis. So now let me draw that for you. Okay, you'll see a cartoon inside your textbook in a second. But so again, here's our structure. I'm going to draw one on one side. We have our ureter traveling out. Right? And remember we had our cortex. We're going to have a filtration system. So I'm just going to draw this thing like this. Tink, 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 tink. And it's going to travel downward, upward, backward, ding, 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 ding. And we're going to now. Okay, so we're not going to worry about the nephron just yet, but we have these papillary ducts, okay, which are going to now feed into the minor and then the major calyces, which will then end up here in our renal pelvis to exit out through our the ureter to go to the bladder. Okay, you can actually see that better. Let's put some lines. No, let's just get out of there. Okay. Let's go here, and you're going to see they're going to have a much prettier picture of what I drew for you there. But look, there's a nephron. It's in the cortex. It's traveling into the parenchyma or the medulla of the kidney here, and you're going to see you actually have this yellow line. That yellow line right there is the papillary duct. It's going to collect things into the minor calyces, which feed into the major calyces, which then drains into the renal pelvis, which is then pulled out through the ileum here. Okay. Now, if we're filtering materials, what I want you to appreciate is the renal artery travels inward, travels to the cortex, will meet that nephron, okay, and then filtration will begin, okay? And essentially what you want to make sure you understand is that the kidneys are an organ of a specifically distinct type of capillary bed we've not seen in this class before. So when we actually encounter the capillary beds, I want you to pay attention to the coloration of the blood flow, because you're going to see an odd sort of portal system. Okay. And number one. And number two, essentially each one of the capillary beds surrounding a nephron. Notice what I just said. Capillary bed surrounding a nephron is responsible for the exchange, figuring out what stays inside the body and what leaves the body. Lots of text on this slide, and I apologize for that, but these are what I consider to be sort of like the distilled, very important ideas. So the blood supply that's actually traveling to the kidneys is traveling with nerve. Remember, just like every other system inside the body. The only thing I could think of that's the exception to that would be, of course, like a nephron, but even then that's just a collection of fluid, not blood. Okay. The kidneys constitute less than 0.05% of the total body mass, but they essentially receive 20-25% of resting cardiac output. Where else have we seen that before? The brain, right? We saw that in the brain, which means you need to pay attention to it. It's going to enter both the right and the left renal arteries, okay? And it's going to branch into this pattern: segmental, interlobar, arcuate, interlobular arteries. Okay, so it's headed towards the cortex is the point that I'm trying to get to here. Okay? Each nephron receives one afferent arteriole. It's going to reunite to form an efferent arteriole. We've never seen this before. Okay? It will divide to form what's called the peritubular capillary bed, and some will have a vasa recta, though some are juxtamedullary. Not what I wanted to do there. J, juxtamedullary nephrons, okay? And what's going to happen, of course, blood flow will then flow out through the peritubular venule to the interlobar vein until it finally exits the kidney itself. The next most important thing to pay attention to here is that it's vasomotor nerves that are regulating blood flow. So when I say vasomotor nerves, hopefully you're thinking sympathetic circulatory system, and that would be correct, meaning sympathetic innervation of the circulatory system, which means it's targeting the afferent arteriole. I should say arterioles, and you'll see that there's a difference when we need there to be a difference. Okay. So let's go here. You're going to see here's that blood flow I was telling you about. Renal artery, segmental, interlobar. Let's jump down to the afferent arteriole here. So that's where we're picking up in this cartoon. Afferent arteriole goes inside this thing that's called the glomerulus of the nephron. And then the efferent arteriole comes out. Notice it's still red, which means all we're doing is lowering the pressure or increasing the surface area inside this capsule so filtrate can come out. Something's going to come back into this, and notice it's going to surround the tubular structure. The tubular structure in this cartoon is yellow, and that is where filtrate is going, and that's the material that came out of the blood. Okay. You'll notice it's inside your textbook they have the peritubular capillaries as purple. That's because it's a mixture of metabolic waste, deoxygenated blood, the amount of time it's spent here, and then it's going to be collected into the interlobular arcuate interlobar to renal vein to get out of the kidney itself. Okay. So before you move on, and before I actually hit the next slide, I would say understand, let's go here. What's Mother Nature doing here, man? She's doing something here that we need to pay attention to, right? That's where your thinking is going to happen. When you're trying to figure out, do I need to memorize this list and this structure? Well, yeah, you could. That's interesting use of your time. But what you're going to discover, of course, is that Mother Nature is going to send this black line here is the sympathetic postganglionic neuron from someplace, right? That's going to regulate blood flow into the glomerulus. So you're running for your life. What's going to happen here? Blood flow stops. You're sitting back, you're relaxing. Blood flow increases. But notice I'm not saying anything at all about the parasympathetic nervous system here. Okay. So the kidney actually has two parts, the renal corpuscle, okay, meaning the nephron has functional pieces. Okay. It actually has the renal corpuscle, which is going to do the filtering of the blood, and then the tubule portion. And you'll see what that looks like in a second. There are distal convoluted tubules that are going to allow for the materials to be collected before it ends up inside this thing we're going to call the collecting duct. I'll show you the entire structure in a second. But pretty much most of your nephrons inside of your body are these things. Whoa, how'd you get there? These things that are called cortical nephrons. Okay, and those cortical nephrons, what they're doing, of course, is they actually have a shorter surface area, so they take less time to filter materials, and we don't tend to use them when things are either really good or really bad. Okay, and what do I mean by that? When we compare them to the juxtamedullary nephrons, these travel deeper into the cortex. Okay, they actually receive blood from peritubular capillaries, as well as this other structure that's called the vasa recta. So we have a modification to the circulatory system here. Probably pay attention to that. And the ascending limb has both thick and thin regions. The purpose of the thick and thin regions, the thick regions, is to stop reabsorption of water. So we're going to create a gradient. It enables the kidney to secrete either very dilute or very concentrated urine. So when we are dehydrated, we use our juxtamedullary nephrons. I swear I do that every semester. I am so sorry. These are juxtamedullary nephrons, right? And they are actually making either very dilute or very concentrated urine. So what do they look like? This figure's inside your textbook, okay? This is the one from the 12th edition. It's probably pretty similar to the one that you have. And if you had all the time in the world, take a look at the vasculature surrounding the tubule structures on the facing you on the left-hand side, right? Okay, and versus the right-hand side. Notice for our cortical nephrons, we create a peritubular capillary bed that essentially collects all of the materials that are traveling out of the tubules. So Mother Nature brings blood in, puts it inside here, forces it out, it travels through the tubule structure, and then she's going to, in mechanisms you have yet to learn, move materials the body needs back into this space where diffusion will recollect the material into the circulatory system. However, in those rare instances when you are dehydrated or overhydrated, right, it is Sunday afternoon, hopefully you've been drinking water all day long, right, you will filter the materials, it will enter into the system here, and now look at the difference in here's the peritubular capillary bed, and then as we travel between the renal cortex and the renal medulla, notice that we actually have something odd there. Nope, there it is. Okay, that makes sense. Good. We're going to have it travel all the way down. It's going to travel all the way up. And notice that there is a thickened portion on both sides here and a thin portion down here. This thickened side here is where we're going to regulate sodium and chloride movement without moving water simultaneously. So we create that gradient I was telling you about. Okay? And all of this is going to be collected in both instances from either the cortical nephron or the juxtamedullary nephron. You are creating urine. Cool. So let's then think about what the tool is that's actually doing the work here. And that is the capsule of the nephron itself. Okay, and what we're going to do is we're going to look at the renal corpuscle here. But you have a glomerular capsule, which has a visceral layer, okay, which has populations of cells called podocytes. Oops, where do we go there? Wait, whoa. You know, sometimes you think you figured it all out. That wrap projections, meaning they're going to increase their surface area centered around the single layer of endothelial cells of the glomerular capsule to form an inner wall of the capsule. Okay? The parietal layer is going to be over the outer wall of the capsule, and what's going to happen is as fluid comes in through the capillaries, it's pushed out between those projections to hit the wall of the capsule, and whatever isn't taken back up into the endothelial cells is going to become our filtrate. Take a look at this here. Blood comes in. We have podocytes surrounding it. See the nuclei of those podocytes here. They allow for material to pass through them. Okay? And whatever's not recollected right back into that efferent arteriole now travels into this thing that's called the proximal convoluted tubule. Notice that's a new piece of information. It means fluid moves right in here, and all of these projections are increasing surface area so we can reabsorb materials. So here's where, it's getting close to our 20 minutes or so, right? So anytime Mother Nature needs something and it's flowing right over the surface here, the cell will collect it and bring it right back into this space. And if you remember properly, there's the peritubular capillary bed. So that's going to now reabsorb that as well. So it's on its surface, it seems like an inefficient system, but it works really well because along the entire tubule structure, which I'm just going to put a capsule here, capsule. Let's go proximal convoluted tubule, descending limb, ascending limb. Let's go distal convoluted tubule and collecting duct. At specific locations, you're going to learn where Mother Nature selects what stays inside your body and what doesn't. Okay, so what we've covered so far is the introduction of where we find our kidneys, what our kidneys look like, what the vasculature of the kidneys looks like, and now we're going to finish up with me pointing out to you again, look, here's nervous system innervation of the afferent arteriole as blood comes into the capsule, okay, to be filtered, and then we're going to pick up when we will stop now.